Hi, this is Qifang Xu from Fox Cancer Center. My talk title is ProtCID, a data resource for hypothesis generation of protein biological assemblies. ProtCID stands for Protein Common Interface Database. So this is the website dumbrack2.fcc.edu ProtCID. Many proteins function as interacting with other proteins, form oligomers in solution. These oligomers are biological assemblies. Biological assembly can be observed from protein crystals in the PDB. Protein crystals have different crystal forms, so we can apply symmetry operators on asymmetric unit to build crystals in Pfam. Here are three distinct crystal forms for human sulfotransferase proteins. It is known human sulfotransferases are dimer in solution. There are many interfaces in these crystal structures. So which one is the correct one? Next group, they use various experiments to discover that this small dimer is the sulfotransferase dimer in solution. The interfaces contains only 10 residue pairs. In ProtCID, we define an interface as the two chains with at least 10 amino acid pairs with C-beta distance less than 12 angstrom and one atomic contact with distance less than 5 angstrom. So in order to identify common interfaces, we use the evolution theory in crystallography, and it basically it says interfaces occur in homologous proteins, and several distinct crystal forms are likely to be biological relevant and part of biological assemblies. So in order to group interfaces into homologous protein groups, we use protein family database, Pfam. A Pfam is a hidden marker model from multiple sequence alignment. We use Pfam, chain Pfam architecture to define a protein chain in ProtCID. For example, this protein chain consists of 3 Pfams. So the Pfam architecture is the Pfam names in the order of the sequence. Our previous benchmarking study shows that if an interface occurs at least 5 crystal forms and the sequence identity is less than 90%, so it's likely to be biological one. In order to identify common interfaces, so we need cluster interfaces. The slide shows you the procedure. First, we classify the PDB structures into different groups. For each group, there are several crystal forms. For each crystal form, there are several structures and their interfaces. Then we apply a hierarchical clustering methods on these interfaces and get interface clusters. For the sulfotransferase case, the biggest cluster actually is the biological interface. ProtCID database contains all types of protein interaction in the PDB. So the first one is chain-chain interfaces. So chain-chain interfaces are grouped by their chain Pfam architecture pairs. Next one is Pfam domain interfaces. Domain interfaces are between individual domains, so they can be intra-chain or inter-chain. For example, the interfaces between SH2 and p-kinase tyrosine. So this interface is intra-chain, missing a chain, and this interface is inter-chain. The next one is about Pfam peptide interfaces. So this shows you the MHC peptide interfaces. The last one is about Pfam nucleic acid and ligand interactions. So this is This is for p53 and DNA interfaces, and this is for interactions between kinases and ATP. Previous ProtCID only contains chain-level interfaces. Now we extended to domain level. Domain-level cluster data greatly increase the number of large protein interface clusters. So I give you an example about the domain cluster. ACT domain dimer. ACT is a structure motif. Is important in the control of metabolism and other cell functions. So ProtCID has an ACT cluster. So you can see from this figure, ACT forms the same dimer in different chain Pfam architecture and different proteins. Jeff Group, a research group at Fortis Cancer Center, this studied phenylalanine hydroxylase PAH. PAH has one ACT domain, and it forms tetramer in solution. After added phenylalanine, ACT form a dimer to activate PAH. So this dimer is same as the dimer in this cluster. So one purpose of ProtCID cluster is to extend the dimers with established biological activities to other family members whose crystals contain the same dimer. So our hypothesis is that, you know, this ACT dimer can activate the other proteins like PAH. My topic is about generate hypothesis for biological assemblies. So I show you 4 homodimers. The first one is ErbB asymmetric dimer. In 2006, Zhang et al. They did mutation experiments and discovered EGFR is activated by this asymmetric dimerization. ProtCID has a cluster about this asymmetric dimer. It contains 4 ErbB proteins, EGFR, ErbB2, ErbB3, and ErbB4. So because not all of the structures of ErbB proteins has this asymmetric dimer in their crystals, so we did data analysis, found that 103 out of 107 structures containing this dimer have active kinase in both domains or in the activated domain. But on the other hand, 50 out of 51 structures that do not contain this dimer consist only of inactive kinase domains. So this cluster presents a good example of how dimerization may be associated with functional changes. The second one is an activating homodimer for tyrosine kinases. This is the biggest cluster for T kinase tyrosine Pfam. It consists of 9 different kinases. For some kinases like BRAF, RIPK2, and RIPK3, different research groups, they did various biological experiments to verify that these 3 kinases can be activated by this dimerization. Some kinases in the same cluster, there is not yet experimental evidence, but this cluster allows us to form hypotheses about their dimerization structures. The next one is about RAS alpha 4 dimer. RAS proteins are HRAS, KRAS, and NRAS. RAS dimerization is essential to activate RAF kinase. Different RAS dimers are proposed. ProtCID has 2 decent clusters for RAS dimers. One is for beta dimer, and the other one is alpha 4-5 dimer. Beta dimer cluster contain HRAS and KRAS, but HRAS only have 1 crystal form. For the alpha-4,5 dimer cluster, HRAS has 7 crystal forms for 91 structures, and the cluster also contains KRAS and NRAS. From literature, the changes on alpha-4, alpha-5 region of HRAS and KRAS can disrupt RAS dimerization, and computation analysis by MD simulation also shows NRAS can form this alpha dimer. So we proposed this RAS alpha dimer is a biological one. The last homodimer is about bromodomains. Beta proteins, there are 4 beta proteins. Beta proteins contains 2 bromodomains, BD1 and BD2, then followed by motif B and ET domain. Bromodomain specifically binds histone and acetylates lysine residues as transcriptional activator. BD1 can form dimer in solution, and beta proteins dimerized through this conserved motif B. ProtCID has a cluster for BD1. It is a head-to-head dimer, but most of BD1 structures do not have this dimer in their crystal. ProtCID have a cluster for BD2. All of the BD2 structures have this dimer in their crystals. This is a head-to-tail dimer. We superposed BD2 peptide interface on this dimer and measured the distance between 2 lysine residues. The distance is 30 angstrom. And this is a typical distance between lysine residues in histone. Since the BD1 can form dimer in solution and the beta protein can dimerize through this motif, so this greatly increases the concentration of BD2 in solution. So BD2 could form a dimer if such dimer exists. So ProtCID provides a very strong candidate. We developed a concept, professional peptide binding domain, PPBD. In order to define a Pfam domain as PPBD, it must satisfy the following criteria. So the first one is the primary function of a PPBD is to bind peptides. We also need structural evidence, and the binding must be conserved. And repeat domain and catalytic domains are excluded. This slide shows you 3 examples for PPBD. The first one is kinase and its peptide interface cluster 1. The middle one is for trypsin, and the cluster is between trypsin and the hydrogen peptide. And the last one is for SH31, and this is the peptide cluster. And one important function of ProtCID website is user can input a list of Unicode codes and get the interactions between them. So we use P53 and input P53 and its interactors on Qifang website. On ProtCID website, ProtCID return a cytoscape pictures about all interactions in PDB. So from this picture, we can see that P53 interacts with other proteins by their PPBD. Study shows that a peptide segment of P53 C-terminals binds to 1433 proteins, but there is no structure. So a potential interaction is between between P53 segment and the PPBD domain 1433. ProtCID provides two sets of coordinates for Pfam-ligand interactions. So the first set coordinates is for each ligand, ProtCID provides all interacting Pfams. For heme ligand, heme can interact many Pfams. And the other set coordinates is for each Pfam, ProtCID provides all interacting ligands and clusters. So this shows you the interaction between pyruvate kinase and ligands, and also their clusters. For Pfam-DNA interactions, ProtCID can provide data on clan level. So the basic idea is that we can model Pfam-DNA interaction in clan level. So the basic idea is that for a clan, some Pfams have DNA or RNA bind structures, but some Pfams do not. So we can model these Pfams without DNA, RNA bind structures by these Pfams with RNA, DNA bind structures. So the top one is for histone clan and DNA bound structures, and these two Pfams can model from these two Pfams. And the bottom one is the DSRM clan and RNA bound structures. So we can model these two Pfams, the RNA bound structures of these two Pfams from Pfam, DSRM and RNA interfaces. I would like to thank everybody in Dumbrek Lab for their friendship and support. Special thanks to Dr. Dumbrek. This research is supported by NIH grant R35, which is awarded to Dr. Dumbrek. This is a screenshot of Zoom Theta meeting of our lab. Thank you for your attention.